we're now going to move into the flash poster session 1. So the speakers, we've got 11 speakers. They are under strict instructions. They've got 90 seconds each, which is no mean feat. So we're going to run straight through to if they can all get into position. So I'm going to announce the first speaker. And then I think they're going to introduce themselves as they go through. So I'm delighted to welcome up to the stage the first speaker of our flash poster sessions, Ryan Marsh. Over to you.
Thank you. Hello everyone, so I'm Dr. Ryan Marsh from Northumbria University, and today I'll be speaking about the baseline findings from the GRAMPUS-CF study, specifically the microbiome arm of the study, because we know that gut microbiome dysbiosis persists today, still in the CF population, despite the rollout of highly effective modulators. And the bigger picture is that people with CF continue to experience persistent intestinal symptoms, for which we think the microbiome plays a fundamental role and may sort of underlie many of the mechanisms involved in these symptoms, which is the purpose of GRAMPUS-CF to elucidate. We collect samples at baseline 6 and 12 months, to look at the longitudinal dynamics of these relationships, but today I'll be discussing the baseline findings only. So what sort of methods have we used? We've used some full-length 16S sequencing, we've done some fancy metabolomics and lipidomics, and we've integrated that with participant clinical demographics to try and form network analyses and look for maybe interventional strategies or approaches or targets, if you like, in these mechanisms. The top-line results thus far are that CF polypharmacy continues to drive the microbiome and the metabolome. Things like oral antibiotics impact the gut microbiota. Obviously, CF comorbidities also impact the microbiome and the metabolome. And if we pull everything together at baseline, we can start to visualize these interactions of particular interest in such networks like I've described here. There's a nice snapshot of that zoomed in where we can see bile acids and particular bacteria relating. And if you want to find out more about this or you're interested, come and find me at poster 1A and we'll discuss further. Cheers.
Hi, my name's Jamie. I'm from Northumbria University, studying under Chris van der Gast. My poster is number 2A and is titled Investigating the Impact of Respiratory Antibiotics upon the Gut Microbiome of Children with Cystic Fibrosis using a high-throughput in vitro model. So for a brief background on why we're doing this research, previous work by a lab group highlighted that gut microbiome dysbiosis persists in people with cystic fibrosis despite the use of highly effective modulators. Others have associated the use of antibiotics with the delayed maturation of the gut microbiome in children with cystic fibrosis. We aim to address the impact of antibiotics on the gut microbiome by using in vitro gut microbiome models. So to do this, the first year of my PhD, I've established a gut microbiome workflow. This includes a storage solution for the samples that we'll be collecting, and then we also established our gut microbiome models and then methods for the analysis of the gut microbiome models. Here's just a brief slide of some of the results we've got so far. If you'd like to hear more about our future work, please come find me at poster 2A. Thank you very much.
Hi, I'm Charlotte Johnson, a PhD student working on the GRAMPUS project. I'm looking into intestinal permeability and barrier dysfunction in people with cystic fibrosis. We've been able to deduce that people with cystic fibrosis have increased barrier permeability, causing leaky gut and an increased inflammation. We have taken this finding and tried to counteract the intestinal barrier permeability and subsequent inflammation by adding a wide variety of anti-inflammatory novel drugs, including HSP-derived peptides, FMT, microRNA lipid nanoparticles to a variety of systems. In this slide, I highlight how Jusfinza, the HSP-derived peptide, significantly increases barrier permeability with the addition of it in both FTSE-dextran flux readouts and electrical resistance readouts or TEER measurements. This is alongside anti-inflammatory lipid metabolites and high expression in superdonor FMT, CAF trio and microRNA lipid nanoparticles, which have also shown a cytoprotective effect against Candida lysine, C. diff toxins and DSS. In addition to these findings, I did confocal microscopy, and you can see that there's tight junction and cytoskeletal adhesion disruption in the cystic fibrosis control compared to its control. And finally, I've used an emerging emulate system to do a bilayer, a multi-bilayer function assay, which also shows decrease in barrier permeability in cystic fibrosis. And if you have any more questions, find me at poster 3A.
Good afternoon, everyone. I'm Darren Sills, a research dietitian from the University of Nottingham. I'm part of the Grampus project as well. So we've already heard people with CF are now thriving rather than just surviving with CF, but gastrointestinal symptoms are a huge burden still, and modulators haven't really tackled that wholly for us. My project is looking at the impact of diet on those symptoms, and the diet has significantly changed in people with CF. The first part of Grampus is to characterise the symptom phenotypes that people have, and so far we've characterised that there may be a mild, a moderate constipation, a moderate diarrhoea and a severe group. And my early data exploration is around the use of dietary patterns and nutrients that people with CF are eating, and I think as we can see the line in the third column, the severe group, the diet does look different. Next is to explore whether that difference is statistical and clinically meaningful. The panel on the left-hand side of the screen is an amalgamation of diet quality and the impact of diet on inflammation. So the DQI is a diet quality index, and the DII is a dietary inflammatory index. Red is poor, red is good, blue is poor. So the severe group tend to have a more severe inflammatory impact and poor diet. Moving on with this one further. Thank you.
Hello everybody. Please indulge me with this slide. We were encouraged to be creative and this is where my mind went. So I've had a checkered history of assembling flat pack furniture over the years, and I think the things that have stood out to me have been, one, the number of spare pieces that you end up with, hopefully spare, but also that actually instructions and following them is really, really important. So keep hold of that. I'm Dan Beaver. I'm a PhD student at the University of Nottingham. I'm one of 4 working on the Magnify research programme, which Laurie's already talked about. So obviously lung MRI has got really significant potential and it is relatively intuitive, but there's also a place for supporting people to understand it as very much an emerging technology. You know, and you need to provide information with people to support that, very much like you need your instructions when putting together your flat pack furniture. So my work's largely been qualitative, and I've been looking to develop an online resource to support the communication and understanding of lung MRI results. I've worked with people with CF, carers and healthcare professionals to produce a number of different resources, and it's a privilege to be able to talk about them today. I've just encouraged people to come along to our poster 5A. If you're accessing this online, then you can scan this QR code and access a digital version of this. And yeah, would be really keen to speak to people and get your thoughts. Thank you.
Hi everyone, I'm Demi and I'm part of the Magnify project, and I'm going to be talking through some preliminary data looking at the longitudinal changes in lung function in CF. So FEV1, or forced breathable volume in 1 second, which is used to monitor people with cystic fibrosis, has been improving with the use of modulators. However, this doesn't necessarily rule out the presence of lung disease. Therefore, we have used Xenon MRI to assess the regional distribution of ventilation within the lung in 3 dimensions. With Xenon MRI, you can visually see ventilation abnormalities, and you can quantify them with a ventilation defect percentage, which is the proportion of the lung without ventilation. So a cohort of patients who were previously assessed in 2018-2019 were reassessed approximately 5.5 years later using Xenon MRI and pulmonary function testing, with all but 1 patient starting modulator therapy in that time. So the results indicate that FEV1, like many studies, improved, which is great. However, despite these improvements, ventilation defect percentage didn't change, and many patients still had ventilation abnormalities on the scans, with those with large ventilation defects at baseline still having large ventilation defects at follow-up. To conclude, Xenon MRI could provide insight into the stability of lung health in cystic fibrosis. For more information, come see me at 6A.
everyone. I'm Eve and I'm also part of the Magnify group. And I've been exploring the changes in airway structure during relaxed breathing using proton MRI. So the motivation for this investigation came from looking at ventilation MRI. And as you can see on the left, I've highlighted a couple of ventilation defects where it seems to be reversible upon taking a deeper inhalation. However, on the right, these defects persist regardless of lung inflation volume. So what we wanted to know was, what is the difference in the airway structure during breathing that causes this difference? From this, we can then use proton MRI, where you can acquire the data during relaxed breathing and reconstruct an image at end inspiration and end expiration, and then segment the airways to reconstruct the airway structure at both lung inflation volumes. You can then take measurements at any point in this airway tree and compare them, the difference in the change during relaxed breathing. We tried to quantify this through size and shape, and trying to relate that to whether we can explain the difference between persistent and reversible defects. So if you'd like to discuss more, please come see me at poster 7A. Thank you.
Hi everyone, I'm Jonathan. I'm a PhD student at Manchester, and I'll be talking about my project, which is ventilation MRI-based computational modelling of inhaled drug deposition in the lungs of people with CF. Now, I appreciate that's a bit of a mouthful, so if you understood all those words individually, but not in the order I just arranged them, we're basically taking the very nice ventilation and structural MRI data from the group at Sheffield. We're plugging that into a mathematical model that we're developing to predict from changes in ventilation relative to controls, how that changes where inhaled drugs end up in the lungs, which is relevant for the inhaled genetic therapies that we heard about earlier. So if you're interested in hearing more about how that works, come and see poster 8A. If you're not interested in that, come and see it anyway, because it is more relevant to you than you think, because the model that we've built up is kind of shockingly similar to models of like contaminant tracking in water distribution networks. So if you've ever drank tap water, it is relevant to you. Yeah, thank you very much. Cheers.
Hi, I'm Dr. Sam Humphreys. I am a postdoc working with Freddie Frost at the University of Liverpool, and my poster is towards cystic fibrosis diagnostic validation in unlinked datasets, leveraging UK primary care and registry data. So a bit of background. So as we've talked about a few times, as people with cystic fibrosis are living longer, there's emerging concerns such as cardiovascular disease, but we don't yet understand the risk profiles for cardiovascular disease in these patients. So primary care data sets are well validated for CV risk, but the issue for CF research is we don't know how accurate CF coding is in these data sets. So our project will use SAIL databank linked with the CF registry to develop and validate an algorithm for recognizing people with CF in primary care data sets. And then this algorithm will be applied to CPRD to describe cardiovascular risk factors and outcomes in this population. So some anticipated outcomes of this project include a validated case finding method for these patients, and the first national profile of CV risk in CF patients. So the really exciting thing about this is that this algorithm will be available for others to use for things like cancer, frailty, etc. So if you'd like to know more about my project plan, please come visit poster 9A. Thank you.
Hello everyone, my name is Rebecca Calthorpe. I'm a medical research fellow from the University of Nottingham, and I'm part of the COS-CF research study, which is developing a core outcome set for cystic fibrosis. So right now in clinical trials, similar trials looking at similar research questions often use different outcome measures. And this can be problematic because it can prevent data being compared or combined, for example, in a meta-analysis, and this can lead to research waste. In COS-CF, we aim to define a small number of key outcomes that can be measured in every interventional trial in CF, no matter what the study focus. To do this, we've already completed a systematic review as well as asking over 600 people from the CF community to find out what outcomes are already being used and what's important to people. And now we're at the point of launching a 3-round eDelphi to finalise what should be included in the core outcome set. But to do this, we need everyone in the CF community to get involved. So please come and visit me at my post to 10A, as well as visiting our website and following us on social media. Please support us by attending a kick-off event planned for later this year. But probably most importantly, please contribute and have your say. Tell us what you think is the most important outcome that should be included in our core outcome set for cystic fibrosis. Thank you so much.
Hi, so my name's Amy Downs. I'm working at the Brompton, and I apologise. Some of this may look a little bit familiar. This is the Matriarch CF study, which is a study of females with CF throughout pregnancy and postpartum and follow-up of their offspring. So this is the Matriarch study that I know we've already heard somewhat about, but just to kind of delve into the 3 parts of this, most of my work will be on mama. My colleague, Dr. Eden Bacobso, is in the audience. He's the paediatrician who's doing a lot of the work on mini and midi. And the way that we've structured this study is that mama focuses on the mothers. It's very well named. And it's an observational study of females with CF throughout pregnancy and parenthood. So we're aiming for a total recruitment of people from preconception throughout pregnancy and throughout the first 2 years, if possible, of that parenthood journey. We're looking at CF outcomes, so lung function, weight. We're looking into the psychological health at all of these 3 stages. And obviously, if we're recruiting preconception, we're aware that we don't know how that journey is going to go, and it's really important that we capture that contemporaneously. And we're doing that through capturing the qualitative experiences as well, which we will do at different time points. Mini and midi is the offspring, and that can be of any parent, biological parent with CF, so males or females. There is obviously a focus on ETI exposure, but not exclusively. We will know which of these babies were ETI exposed in mini, and that will be infants recruited from birth until they're 2 years old. And we'll look at things like liver function through that time period, sweat chloride, what their exposure to modulators is, both in utero and through breastfeeding, and the overall development. And mini is a cross-sectional cohort study, and we're going to capture the cohort we have of children aged 3 to 6, particularly those that were ETI exposed in utero. There's the capability to do LCI and MRI for lung development, and Imogen kind of referenced the importance of that earlier, and their developmental milestones as well. And although we hope there will be people who end up recruited possibly as a family into all 3, they are independently running as well to kind of maximise recruitment. Thank you.